Hello, this is Dr. Esterline in the chemistry lab. I am going to demonstrate lab number 3, identifying organic unknowns for all of the chemistry students taking Chem 104L. So to show you what I'm talking about, if you have your lab manual, pull it out and you can kind of follow along. So we're on lab number 3, identifying organic unknowns. If you were in lab, which canceled because of bad weather, you would have been given an unknown and you would have taken it through all of these tests. So what I'm going to do is I'm going to take you through these tests myself with unknown A. So you all have unknown A. We're all going to use the same one since I'm doing the lab. I did post videos above this video that shows very well put, much better videos than mine that shows the bromine test and what's positive, feelings, 2,4-dinitrophenylhydrazine, which is called 2,4-DMP for short, chromic acid test. I didn't post one for water solubility, but we'll talk about that and try it. Okay, so what we're basically doing is we're adding various chemicals to different organic substances, and depending on how they react, we can tell whether there's a presence of a functional group or not. So the possible unknowns is hexane, cyclohexene, formaldehyde, heptanal, ethanol, octanol, acetone, cyclohexanone. Ignore these numbers here. This is the shelf number that they're on in our stockroom. Your unknown is one of those. So part of your goal is to use the data that I'm going to show you here in a second to figure out which one of those is unknown A, right? And we're going to take it through the chromic acid test, 2,4-DMP, feelings test, and nasty bromine that will burn you. I'm going to try it in these dimple plates, and if it doesn't work out well, I'll do it in a test tube. The advantage of these dimple plates is I can use very small amounts, and it's a white background, so we should be able to see color changes or solids that form. So before we start, I also posted this data sheet that Professor Wino put together. Thank you, Professor Wino. It lists all of these possible unknowns on the left-hand side here. They're structures. So she looked them up for you. You're welcome. The first one is hexane. The second one is cyclohexene, formaldehyde, heptanal, ethanol, octanol, that's acetone, or we can also call it 2-propanone, cyclohexanone. So those are really good ones for you to practice. In Dr. Banzal's lecture, you need to know how to name these types of things. So good practice for you. We're dealing with simple structures. Some of them don't have any functional group at all. It's just an alkane. Some of them have a double bond, so we say it's unsaturated. A carbon-carbon double bond. Some have a C double bond OH, which is an aldehyde, and then a longer carbon chain aldehyde, an alcohol, longer carbon chain alcohol, ketone, and ketone. So the reason why we're dealing with 2 of each for these is it has to do with solubility rule, which we will test our unknown and see if it's soluble in water. So I'm not sure exactly what Dr. Banzal tells you in class, but textbooks that I have used kind of break it down like this. It depends on the number of carbons in your structure per polar functional group. Polar functional groups are things that have oxygen, things that have nitrogen, and halogens for the most part. Chlorine, bromine. Okay, so in our examples here, if you got an oxygen, you got a polar functional group. If you got a carbon-carbon double bond, that is not a polar functional group, right? Or an OH, polar functional group. So if we go through these, if you have 4 to 5 carbons for every one polar functional group, then we say it's slightly soluble. In general, the organic chemical tends to be slightly soluble in water. What does that look like when we test it? Well, it'll look cloudy. If it has less than 3 carbons per polar functional group, then it's going to be water soluble. It's just going to dissolve and look like water, one layer. So cloudy, 1 layer. The opposite would be, it's more than 6 carbons, 6 or more, per polar functional group, so it does not dissolve in water. It is water insoluble, and we'll see 2 layers in the test tube. And I should have prepped a couple of test tubes ahead of time just to show you. I'll just do our unknown right now. So I apologize, I got to move you around with me because you're in my left hand as I'm working with my right hand. So I'm working as a one-handed chemist. I'm going to add some of unknown A into a test tube. And then I'm going to add water. All right, so hopefully you can see that. Looks like 1 layer. Let's add some water. Go to the sink, turn some water on, see if I can catch some of it. And what you should see is, yeah, there's some bubbles there from the chemicals mixing, and it all becomes just 1 layer. It all mixes. So we can say that our unknown is, I'm kind of waiting because you can see the bubbles kind of settling out. It's definitely 1 layer. So if it's 1 layer, that means water soluble. It must have 1 to 3 carbons per polar functional group. I'll let you figure out which ones of those have 1 to 3 carbons per polar functional group. Let me show you an example of what would have happened if it had more than 6 carbons. So let me grab this hexane here. Once again, I apologize. I'm flipping you around all over the place as I grab things. I didn't do this in advance. I'm going to pour out a little bit, yep, all over hexane. And if I put hexane in that same test tube that we just added water, watch what happens. I don't know if you can see that very clearly, but there is a line. There's a line separating the hexane on the top here, and water on the bottom, right? Most organics tend to be less dense, so they float in water, just like a cork floats in water. A few that are like highly halogenated, they would sink in water, but definitely you see 2 layers there. So hexane, because it has no polar functional groups, right, it does not dissolve in water. So we've ruled out 1 possibility for your unknown. Let's do the bromine test. So I'm going to take some unknown and I'm going to add it into this dimple plate. And then I'm going to add a little bit of bromine. I'm doing this in one of the hoods. I'm going to step back and show you. In one of the hoods in the organic lab, just down the hall from where you normally work. Because this bromine is nasty stuff. I've seen a graduate student when I was in graduate school get burned severely by this. So I'm always very cautious about bromine. Okay, so I'm going to add a few drops and you observe what happens. Did the red-orange color go away? Well, let's double check. I'm going to mix it and see if it goes away. Did it go away? No, it did not. It did not go away. It stayed orange. So for the bromine test, back on this table, if it goes colorless, then it has a carbon-carbon double bond. Well, ours stayed kind of this red-orange, right? It's kind of diluted down red-orange because the bromine's diluted down. What would it look like if we had a double bond? Well, do I have a chemical that has a double bond? Cyclohexene right here. So once again, apologies, I did not prep this ahead of time. Gotta go grab a beaker. There we go. Now I gotta one-handedly manhandle this giant jug of cyclohexene. We just need a little bit. Come on, a little. Come on. There we go. That's good. I didn't spill it that time. Yay. So I have some cyclohexene. It has a double bond. What happens if I put some cyclohexene here that has a double bond and then we add bromine to it? Well, we should get a positive reaction, right? That double bond should react with the bromine. Let's see. crossing my fingers. Yes, do you see it? It's going away. Here, I'll add it again. 2 drops. 2 drops, yeah, so 2 full drops. I'll even mix it. You can see that color going away. That would be a positive test for cyclohexene. Your unknown, it stayed kind of this orangish red. It did not go away. So that's the result for the bromine test. Stayed red-orange. Right, I know on the sheet it says red, but red to orange would be a negative test for a double bond. Going colorless, like this, is a positive test for a carbon-carbon double bond. Cyclohexene has a double bond. Okay, moving on. Feelings test. The feelings test is the next column. Man, my phone keeps going to sleep. It gives a dark red solid if you have an aldehyde or if you have a ketone. Nope, sorry, just aldehydes. Just aldehydes. So we're looking for dark red solid. Now, I don't know how well this is going to work in this dimple plate. I'm putting a little bit of, I got to add feelings A, feelings B. Feelings B has a copper ion in it, so it's kind of a pretty blue color. And then I'm going to add unknown. Right, going over and getting some of the unknown, adding that to the feelings reagent. So this one, I think this one you have to heat for a little bit. So that it may not work in this dimple plate. I'm gonna mix it. Do you see how it kind of stays a blue color? I'm not really seeing any red forming in there, but we'll come back to it and check it in a minute. See if it turns red. Yeah, so I'm kind of adjusting the procedure a little bit. And this third one, right, so I don't see dark red. I'm not seeing it. And this third well, I'm going to do the 2,4-DMP test. Dinitrophenylhydrazine is what DMP stands for. It's positive for aldehydes and ketones. You get a colorful solid. So I'm going to add a little bit of the DMP. I can see my unknown I put in here ahead of time has evaporated. So I'm going to add more of the unknown. And this one may be one of those where you have to heat it a bit. I'm going to use the other end of my stirrer. And once again, we may have to come back to that one. And I may even have to redo it in a test tube. Right, so it's kind of just the color of this 2,4-DMP solution right now. Okay, so we'll come back and check that, see if there's any solid in there. Chromic acid test. Chromic acid test starts out with this kind of orangish dark color. But it says the chromic acid test should turn green to blue or maybe even blue-green for aldehydes, so the 2 aldehydes and the 2 alcohols. So it's positive for aldehydes and alcohols, turns blue-green. Let's see what happens here. Okay, so this one normally you can see it almost immediately. And I'm not seeing any blue-green in there, are you? Well, let's do what I haven't been doing since I did the bromine is, let's do one that's definitely positive. Chromic acid is positive with aldehydes and alcohol. So let me grab some, what do we got here? We got some heptin now. I've got heptin now. I don't know if I can open this bottle with one. I'm gonna sit you down for a second, sorry. There, got the bottle open. Heptin now. Let's do, right below this test, some heptanal and see if it turns blue-green. So there's the heptanal. See, there's no color in it. All right, make sure I grab the right bottle. Does that say heptanal? Yep. Okay, so now I'm gonna add chromic acid to it, see if we get any blue-green. Not working like I would expect it to. Yeah, so this is challenging to run these with me recording by myself, holding this and doing this lab at the same time. Hang on a second. Do I see some kind of reaction? Possibly. I'm gonna grab another stirrer. I'm kind of seeing green color. I don't know if you can see that or not, but a little bit around the edges. Yeah, can you see that? Right up here around the top? That's kind of blue-green. Now, probably if I used a different aldehyde or alcohol, it would have been even more green than that. In fact, I'm tempted. I'm wanting to grab another one and test it. Blue-green. We need